So why I'm doing what I'm doing, which means biology, a little bit of physics, computation, and why this is good for you. So for that reason, I start explaining to you why do we need system biology here. You see, this is a keyword. And then, should we cross out computation or not from biology? This is, and then you respond. I hope talk with me about what do you think? Should we cross it out or not? But first of all, independently of your choice to do computation or not, I think that systems biology is quite an important idea that you need to understand as soon as possible. So, what I'm going to do first time, I want to show you, I hope this is connected with the internet. Yeah, good. So you see, this is the first department of systems biology in this country, created about 5 years ago at Harvard Medical School. And this map is the metro at Harvard. So you can take, for example, this line to go from MIT to Harvard, so they. I encourage you to write down that at Harvard you have a systems biology department, and this is on the web page, and you visit it. Now let's see, for example, what type of PhD programs, because if you want to go to PhD programs, you have to see what type of programs do they have. So. You see biomedical engineering, applied mathematics, applied physics, chemistry and chemical biology. So everything is there, you see? Chemistry, biology, applied physics, mathematics, you just saw. So all this combination together, it's part of this systems biology. So of course, if you like more biology than computation, you go towards more biology. But your next door neighbor will be maybe a mathematician, or the other one maybe a chemist. So it's going to be a very interesting environment for you to be every day. So also interesting, let's see if I take this out. If you go backwards, the head of the department, he created this. He was hired exactly for this purpose, to create this new department. And then you see, just go by step, and you can read about the CV of every single faculty here and see what they are doing. And for example, this person here is doing computational biology, and I know for Dr. Pamela Silva, I know her, she's doing like transport from inside cell, from nucleus to outside, and she loves cats. All right, so do you have questions about first part? It's about I want to introduce you to this new systems biology department. Okay, it's not the only one. There are many other universities that offer such an interesting program. Now, my second goal is to explain what is this systems biology, and I'm going to use another website which is from an Institute of Systems Biology in Seattle. And this is another famous institute. You can go either on the East Coast, as you see, or on the West Coast. And they use a very interesting approach to explain why do we need systems biology. And I recommend you to write down this keyword, Institute of Systems Biology, Washington, Seattle, and check the webpage. You will learn a lot. So it's again Institute of Systems Biology, Washington, Seattle. Leroy Hood is the head of that institute. So they have an interesting introduction here for you, and they explain the following thing here. Probably you don't see, but I will read for you in my words. So imagine you have three objects, yeah, which is just a small metal cup a glass, like a balloon, and a piece of wire, a coil. Yeah? Each individual has absolutely no meaning. It's useless. Yeah? What can you do with a piece of glass, a piece of metal, and a coil, right? But if you put this together, what do you get? Some interesting object called the light bulb. So the point of this very, very simple introduction is that parts taken separately are useless. But if you put them together, they are extremely useful. The property of these parts put together are completely different than the property of each individual part, glass, metal, and the coil. Now, this is exactly this trend. It's opposed to the trend taken in biochemistry or biology in the last 40 years, when you take cell life, split it in so many parts, and study each individual piece, right? Each individual gene. And this was very good for the development of molecular biology and biochemistry. But now it's time to put all these parts together, to take the glass, to take the metal, to take the coil, and understand the bulb, get the light. And there's the reason this word systems is in that big title. Because the bulb is a system composed of these three different, unrelated pieces of parts, okay? Then they go further and say, all right, what can you do with these bulbs? Take bulbs and wood, like batches of wood, what you can do with this? A baseball or a soccer event. You put people on the benches, you light, and everybody's so happy there and looking at the ball. So you see, it's a different level of a system. So once you understand simple systems, then you go and understand big systems. So now what's the difference? So it's another important keyword here. It's called emergent properties, which means that these properties were not part of those parts. So the light, the bulb, as function, is not part, this property is not part of the glass itself. And this part, the emission of light is not part of the metal itself. It's only when this system comes together. All right? So I hope I convince you to, whenever you have time, to go to these two sites, search and find what is systems biology. Then, the next part is to show you, of course, what I'm doing, and then it's an example of a system in biology. Then you have the gene regulation and cell proliferation, apoptosis. All these things are governed by many, many interacting molecules. If you take one piece out only, let's say you study only this cytokine here, this is like studying only the glass out of the bulb. Breaking the glass and take a piece of a glass, it's transparent, it's beautiful, right? But you don't study the bulb and the function of that. Only if you combine this cytokine with the entire molecules in a very specific map and interaction, then you are able to understand the function. So that's the goal of this system biology, to understand this complexity. Well, once I'm here at this point, I mention interaction, right? This cytokine interacts with the receptor on the membrane. How should you understand this? Only at the level of coupling, or should you understand at the level of concentration levels? And this, as you just mentioned, the concentration level are so important, yeah? As a consequence, for that reason, you need computation. Because you need to find out what's the level of this cytokine here in order to open an entire pathway to get to cell proliferation. Maybe it's not enough, that concept. Maybe it has to be a threshold. Maybe it's can be linear. Maybe it's abrupt. It depends on temperature. So for that reason, we need computation. Now, my slides are not having computation at this point. So what I'm going to show you, what I did with my students over the summer. We actually studied, we collaborated with Dr. R.J. in biology, and we studied a gene which is controlled by iron. So you know that iron is part of metabolism of bacteria. They enjoy iron, yeah? I enjoy cookies, they enjoy iron. So if you take the iron out of that solution, they got in panic. And they opened some gene, which is called IUCD, and they said, whoops, we don't have iron, we have to go and search for iron. So then the question is, what is the mechanism of this iron regulation? So, how to check for this iron? So the idea is that if you use a reporter, a GFP, green fluorescent reporter, and if I'm not mistaken, by the way, they got Nobel Prize for chemistry, I think exactly for this green fluorescent protein 3 days ago. And what you do, you take this gene fluorescent protein as a reporter, hook it with that promoter from iron consumption, which is regulated, and then when this bacteria open that pathway for iron searching, they actually open this GFP production, and you see they start to light. So this is real bacteria. We measure this with a confocal microscopy down here in our suit with a microscopy. And this is real bacteria, and they shine because they lack iron. Now, the interesting part is the following. If at some point in time, usually in the morning, you deprive them from iron, this is a step shock of iron. So you change the media, kill the entire iron out of that media. Then what you see, this GFP starts to grow, accumulate, grow. So all these bacteria starts to work and search iron. But you see there are quite different stages in this iron controlled pathway. So what are we interested in are two things here. How fast is the response of this bacteria to iron shock? And then what happens in this region when for some reason it flattens out? So I'm going to show you 3 pictures of bacteria, one taken from this region, one from this region, and then one from later on. So 1, 2, and 3, all right? And you'll see that initially the bacteria is small and just a few bacteria, the colony is at low density. After about 180 minutes, they start to divide, and for that reason, we believe that plateau in that GFP accumulation. And then later on, they become again small, and then something is happening. We don't know, so this is one of the way to correlate the iron, the GFP accumulation with the phenotype and what's happening here in bacteria. And we use microscopy for this. And another avenue of research is to find, as I said, the meaning of this GFP accumulation. So if you give them a little bit of iron, this is quite interesting. If you take the iron completely, this curve, this is a blue curve, 0.00 micromole of iron, no iron. GFP is growing. However, if you give them a little bit of iron, 0.02 micromolar, just a little bit, it's huge. It's a growing GFP. Why is that? So probably it's a balance between metabolism and promoter activity. If the iron is not present, the promoter is open and the bacteria is searching for iron. However, they need iron to produce energy, so they don't have enough energy. If you give them just a little bit, they have, okay, you have enough energy, but also they are in panic and they are looking for the iron. If you give them a lot of iron, like you see, this light blue is nowhere. You cannot open any promoter. The bacteria is okay, I have enough iron, I don't care much. So there is an optimum amount of iron to give them to open that promoter. And you'll see this here. So one of the goals of the future research is to find out exactly this iron concentration versus bacterial response. It's like this. Give them a little bit of iron here, this huge explosion of GFP, and then it's going down, and not much on this side. So usually in this type of maximum and then minimum, it's quite an interesting phenomenon. In physics we call it resonance. In other we call it in mathematics just a maximum. However, what is a mathematical model that you can create to explain such a sharp maxima in the response of bacteria to iron concentration? I don't know. I hope we can figure it out. And probably this is a response for many other environments. Just give them a little bit and they'll respond with time. Okay, so this work was done with Alex and Adam and presented here in September. More details on this poster, but for this short talk, the idea is